Dr. Hanson, I first met when I was a graduate student. He was kind enough to serve on my graduate committee, and he's always been one of the scientists that I've really looked up to as a role model, and just an excellent scientist and just a great man. Dr. Hanson was a BYU student back in the 1960s. He went to UCLA to their dental school and got a DDS. Then he went to graduate school at the University of Utah and got a PhD in pharmacology. He then did a postdoc at NIH and then has been at the University of Utah at the Department of Pharmacology and Toxicology for about 30 years now. He was asked to serve as the director, the acting director of the National Institute of Drug Abuse, one of the NIH institutes, and he did that for a couple of years, about 10 years ago. Please join me in welcoming Dr. Glen Hanson.
I'm going to come out here in front. Is that all right? Okay. I hate getting behind the podium and I hate standing behind the table. So we're going to be talking about drug abuse because that really is my area of expertise, even though I was originally trained as a dentist. And I actually practiced for like 10 years. I got to the point where I decided that wasn't me. And I went home and announced to my wife that I hope she hadn't gotten used to actually having money. And my wife, being incredibly supportive and a wonderful person, she said, "Sure, that's fine." What did I just do? She said, "That's fine." And I went back and got my PhD in pharmacology. And eventually, through no great planning on my part, I made my way into the area of drug abuse. And that was kind of troubling for my mother, to be quite honest, because she liked me as a dentist. She liked to brag to her friends about, "Oh, my son, the dentist, you know, he does this, he does that." And when I came home and said, "Mom, I'm going to be a pharmacologist," she said, "Well, what's that all about?" And I said, "That's somebody that studies drugs." She just kind of looked at me in disbelief, and one day she introduced me to her friend and said, "This is my son." And the friend said, "Well, what does he do?" And she kind of looked a little baffled and said, "Well, he used to be a dentist, but now he does drugs." So that's what we're going to talk about. We're going to talk about doing drugs, but we're going to talk about it in a way, hopefully, that you can appreciate that our approach and attitude towards drug abuse and addiction today has changed considerably. And that has affected a lot in terms of public policy. In fact, just last week, I was telling Dr. Sudwick that I was testifying before our state legislature on the tobacco tax, trying to educate them about the neurobiology of nicotine and why that was something that they should consider in their policy making. And so science has become the critical factor in policy making and impacts you as citizens. And it's also an important issue that we as members of the Church of Jesus Christ of Latter-day Saints need to keep in mind as we evaluate our family members and ward members and friends who are wrestling with issues related to addiction and drug dependence. So I've titled this New Perspectives, and what that means as far as diagnosis and treatment is concerned. So just to give you a quick overview of what past looks like, and for some people this is still the present for them. They have a hard time getting out of the past. But the past comes because of dependence associated with some of the natural substances that have been around as long as man has been around. These things are in plants, they're easy to access, and people have gotten in trouble with them. The opium products have been part of societies for millennia, not only because of its abuse potential, but its pharmacological potential as an analgesic. It's one of our best pain relievers, pain managers, but it's sometimes difficult to draw that line between using it in a medicinal legitimate way versus getting in trouble with it. And as you've heard, I'm sure, on radio and seen on television, one of the biggest problems we have in the state of Utah and across the country is prescription drug abuse. And most of that is coming from these drugs that are associated with opium products or opiate systems, as we refer to them, in the brain. So historically it's been there. These problems are still there. At the turn of the last century, as the industrialization of nations took place, these drugs of abuse were taken out of their natural settings and were put into products such as the patent medicines. We've all heard of Coca-Cola. The coca part comes from the coca plant. So originally cocaine was included in this patent medicine, and most of its pharmacology was because of the presence of the cocaine. There were a lot of indications that were ascribed to this product which were unrealistic and not realized in a pharmacological way. But coca made everybody feel good, so it really didn't matter whether it did the rest of the claims or not. People were pleased with the product. Then marijuana also has been around for many millennia. It's a weed, it's a plant that grows almost anywhere. It can be useful because of its fibrous nature. It can be used to make clothing, rope, paper, so it's got some real utility that's been important to societies. But when you start to smoke it, then it's got some pharmacology that can be addicting and still creates problems. And then, of course, the alcoholic beverages. Alcohol has been part of societies forever. And as we became more sophisticated, we were able to distill it and to purify it and increase its potency. And with that, we increased the problems of addiction and dependence that it caused. So these are many of the same dependence and addiction problems we wrestle with today in the world of drug abuse and addiction. But in those days, we really had no understanding as to what was going on with these drugs, other than they had an effect. They seemed to change behavior. They got people into trouble. But that's about as far as our understanding went. And when we tried to create policies to deal with those problems, they oftentimes were off target. Just as an example, the syringe was originally created in order to try to decrease dependence problems, particularly with morphine. Morphine was a big drug used during the Civil War. It has always been an important drug for wars for two reasons. One is the obvious reason. What would that be? Why would you want morphine for war? A lot of pain. People get their arms cut off and they get shot or stuck with swords, and so it's a good pain reliever. But the second part, which is probably even more important, is it is an anti-diarrheal drug. And one of the biggest killers in wars are things like dysentery. So this drug, which helped to relieve the loss of liquids through the process of dysentery. Well, but it had the dependence problem. Half of the soldiers in the Civil War came home addicted to the morphine that they had been using because of injuries. So they thought if they could inject the stuff rather than taking it orally, they could reduce dependence and addiction, but it turned out to be exactly opposite. By injecting it, the stuff was more potent, its effects were more dramatic, and this caused greater incidence of addiction and dependence. Another part, an assumption was made that addiction was always a part of the undesirable social folks. That people who became addicted or dependent on these substances were usually that part of your community that you really didn't want to be in your neighborhood. So we always would push them to the other side of town or encourage them to move on and go somewhere else. We didn't want any part of their immoral actions, if you will. And obviously now we know that that's certainly not the case, that addiction and dependence problems are everywhere. They're in good families. They're in families that are dysfunctional. It's pretty much ubiquitous. And that kind of comes to this piece here, that those who are addicted, they tend to be obnoxious. And I think that's true. They tend to have behaviors that can be quite dangerous, and that's also true. And that sort of takes you down that road, that judgmental road, that these people are evil and they're immoral. But in fact, a lot of that behavior is a consequence of the drug, what the drug does to the brain and the cognitive and executive functioning of the brain. So these are some of the conclusions that we drew previously. And as a result, we said addiction is a morality thing. It's all about good versus evil. Addicts and dependent people are evil. Folks that don't do that, they are good folks. And so, how did we deal with these kinds of issues of good versus evil? We frequently would turn to our criminal justice system to take care of this. So this was our principal treatment for problems associated with drug dependence. What we would do is we'd put them in jail. And if we wanted somebody not to get into trouble with drugs of abuse, we would increase their jail term. Or we would penalize them or punish them in some fashion, not recognizing that you're fighting underlying biology. We thought, mistakenly, that we were just fighting bad decision making that could be corrected. And I'm not saying that that doesn't occur and that isn't an issue, but in severe dependence, it goes way beyond that. And so jail time or the criminal justice system isn't very good at correcting the neurobiology of addiction and dependence. So where are we today? And that's what I want to focus on for the last few minutes that I have with you. We're in an era of scientific discovery. We know a lot. We probe. We use all kinds of new technologies to help us to understand how the brain works. And as a result, our attitudes have changed along with it. How does information processing occur? How does behavior come about? What does it do? Can you manipulate it? Can you change it? And while the criminal justice system is always going to play a role in drug abuse and addiction because some of the behaviors associated with people who find themselves in that circumstance are behaviors that are dangerous, some of these people do terrible things. They do terrible things to themselves. They do terrible things to their families. They do terrible things to their neighbors. That's going to be criminal justice, but it is not going to be the answer. It may be able to help us to work towards the answer, but it's not going to be the answer. Public policy folks recognize this now. I don't know how many times in this state and across the country when I was the acting director of the National Institute on Drug Abuse, how many times I testified or interacted with the criminal justice system, with the DEA, with the division or the Department of Justice, and talked to them about what should we do. Our jails are filling up with people who have trouble with drug abuse, and yet jail time doesn't seem to resolve it. Once we turn them loose, guess what happens to them? They come right back. And how do we break that cycle if we don't deal with the underlying biology that's going on? So, it looks like a disease. It's got all the features of a disease like diabetes or cardiovascular disease or Parkinson's disease. And so maybe treatment should be considered rather than punishment as a way of dealing with it, and use the same techniques for treating this disease as we use for other kinds of similar types of diseases. Well, what I want to do is I want to identify some of the technologies that have gotten us to where we are today in terms of understanding the underlying biology of addiction and dependence, and establish some basic principles that have resulted from that technology. So how did they change our attitudes, and what impact has it had on our approaches per se? So let's start off with 4 that I've identified, and there are more than this, but I think these are the 4 principal technologies that have really changed the way we think about drug abuse and addiction. And the first one I want to talk about is microdialysis. How many of you are aware of microdialysis? Okay, so there's a few of you that have heard of it. Let me talk to you about what this looks like. It's a relatively recent technology. What it does is it allows us to measure minute quantities of neurotransmitters. Y'all know what neurotransmitters are, right? So these are the molecules, the chemicals that are released by neurons in the brain. That's how they talk to each other. So the neurotransmitter systems are the basis of brain function. If you don't understand neurotransmitters, you don't understand how the brain works. So this is a means of evaluating the release of neurotransmitters in a localized brain region so that we can identify which part of the brain is associated with what kinds of activities. This is what we're looking at. We want to assess these transmitters here and find out what increases or what decreases their activity. And microdialysis helps to evaluate the quantity of these transmitters. So what does microdialysis look like? Well, it's a cannula and it's implanted into the brain. And these are actual microdialysis cannula. On the one end, you've got an infusion piece where you flow artificial cerebral spinal fluid. On the other side, you have an output where you collect that cerebral spinal fluid. And then this fluid flows in down to this tip, and then it makes its way back here through the output. And this tip is a dialysis membrane. And a dialysis membrane is one that allows molecules, depending on their size, to flow either in or out or across that. So by doing this, we can sample the exchange of molecules at this tip where your dialysis cannula is located. Here's an animal where we are implanting a microdialysis cannula. So the animal is anesthetized. We put it into a specific place in the brain. And in order to make sure we get at the exact right place, we use a device called the stereotactic instrument, which gives us the coordinates of going anterior, posterior, dorsal, ventral, and we can place it within a fraction of a millimeter in that brain region that we're interested in analyzing or assessing. This just illustrates brain placement. This is a rat brain. So you can see we can drop that microdialysis. Here's the dialysis part of that cannula in this region, or we can put it in this region. And the length of the dialysis probe is critical in determining what your sampling is going to look like. If we want to be very specific, we make the length very short. If we want to analyze a large area, then we make that dialysis piece long so that it's analyzing a large part of the brain system. Without going into a lot of detail, there is a flow dynamics here. And all that really means is that there are molecules. So here's the artificial cerebral spinal fluid with the dark molecules. As they come around into the dialysis piece, they can flow out across the dialysis membrane, whereas the neurotransmitters, the open circles out here, can flow into that, and they can be collected and they can be analyzed. So we can literally tell on a time sequence what's being released, how much is being released, whether that activity goes up or it goes down. So areas of great interest to us in drug abuse is the systems associated with the transmitter called dopamine. You all heard of dopamine? Dopamine for drug abuse is the #1 neurotransmitter you need to understand. And the pathways that are dopamine related, most of them, the origin, the cell bodies come here in the deep part of the brain, the brainstem area. They project forward into a variety of different brain regions, and dopamine is released up here in the hippocampus. This is a big memory area, the striatum. This is a big motor area that's responsible for behavior development, and frontal cortex, which is important for cognitive processing. But a critical area for us in drug abuse is this nucleus accumbens area. That's the target of the pleasure pathway. That's the part of the brain that sends a signal to you that, boy, that was a lot of fun, or wow, that felt really good, let's do that again. So anything that seems really rewarding, even if you do a test and you get a good grade on it and you feel really good about it, dopamine is squirting out in your nucleus accumbens. So these functions are associated with the dopamine systems: reward, pleasure, motor, compulsion, perseveration, and decision making. And the reason that's important is because the drugs of abuse interact with these dopamine systems. And a lot of what they do comes from the biology of those dopamine systems. So now what I'm going to show you is a microdialysis assessment of a nucleus accumbens in a rat, because they don't want us to do this in humans. You know, they want us to put these probes in human brains, but they will let us do it in rats. So we've got these sticking in the nucleus accumbens of rats. And this is what it looks like. When you give a rat amphetamines and through microdialysis you measure the release of dopamine, you get this nice surge in dopamine, like a 10-fold increase in the amount of dopamine that pleasure pathway releases. If I were to give the rat cocaine, I would see something like a 300% or a 3-fold increase. If I gave them nicotine, I would see something around a 2-fold increase. Ethanol will give something around a 2-fold increase. So not only do we appreciate that dopamine release is being stimulated by all these drugs of abuse, but I can actually see relative stimulation that's occurring. So this one is particularly interesting. This is an area that a lot of my research focuses on, are the amphetamines and methamphetamines. See how much greater it is than any of the other drugs of abuse? 10-fold, and we've actually done some experiments with microdialysis where it went up 50-fold. So that system's being stimulated 50 times greater than what it's designed to handle. 50-fold over what it's supposed to do. And as a result, when you give high doses of methamphetamine or amphetamines to animals, it can kill these dopamine cells. It pushes them so far, it asks it to do something so much more than it's designed to do, that it'll kill the cell or cells. And that damage can be permanent in methamphetamine addicts that have been using very high doses for a long period of time. Now, just to give you some relevant comparisons here with this, if we looked at natural rewards, so these are rewards that are going to activate that system but do it in a natural way within the physiological boundaries of the system, we see food, you know, like a chocolate cake or a pie or a steak or something you really like. It can give you about a 50% increase. Sexual activity can give you about a 2-fold increase. But in neither of these cases or any other natural stimuli can you come close to what methamphetamine does. Same system, but it doesn't come close to what the drugs of abuse do. So what does that mean? Well, from those discoveries, we've made 3 conclusions that are based on good scientific neurobiological principles. One is all drugs of abuse release dopamine in the nucleus accumbens. There is no exception to this rule. Not only that, I could go one step further and say everything that is addicting stimulates the release of dopamine in the nucleus accumbens. That's the reward pathway for the brain. The second thing we know, that in a way it hijacks the natural reward systems, especially if it pushes it, like the methamphetamine, way beyond what it's supposed to do, and it damages some of those neurons. Now they're not available to transmit natural reward information. So these people are crippled in their ability to enjoy natural rewards. And you hear this all the time when you talk to them. They say, you know, life just isn't as exciting and wonderful and pleasurable as it used to be. I don't get the same enjoyment out of the stuff I used to enjoy before. It just isn't there. Why? It's because those accumbens dopamine neurons have been damaged by the use of the drug. And then finally, vulnerability to these drugs seems to be linked to this dopamine system. We find that in people who have a dopamine system that's compromised, in that it's underacting, it's underperforming, they're much more likely to get into trouble with drugs of abuse. Why would that be? Why would these drugs be appealing to these folks? Yes?
They can feel enjoyment.
Yeah, it compensates for this lack of stimulation that's occurring naturally in their brains. And we've seen this in animal models. If we go into an animal and we pre-treat them with something that damages that reward system, and then we give them an opportunity to work to get a drug, they will work much harder to get that drug. It happens in laboratory animals. It happens in humans. We've got some really good data that show that the same kind of thing happens in humans. Okay, the second technology that's helped us to understand what's going on with these drugs is known as quantitative autoradiography. How many of you know what this is? Always the professors that raise their hands. So let me show you what this is all about. This stuff came on board in the 1980s. Really, really exciting because it gave us the opportunity to actually visualize in a brain what kind of biology is being changed by conditions or being changed by drugs, such as the drugs of abuse. So what it is, it's a quantitative assessment, and it's got this anatomical piece to it. It allows us to evaluate the states of things such as receptors. What are receptors? Those are the targets of the transmitters, right? So when you talk about transmitter action, there's two pieces. One is the molecule that's released, that's a transmitter. But the other piece is the site at which this molecule interacts, and that's the receptor. So it gives us a means to assess what the receptors look like, and other proteins known as transporters. And these proteins are important in taking transmitters and putting them where they're supposed to be. So after many of the transmitters are released, they're grabbed by their respective transporters and they're brought back into the brain cell so that they can be repackaged and used again. So these transporters are very important for managing the dynamics of the neurotransmitters. So autoradiography gives us a way to assess these receptors and transporters. And it looks like something like this. First of all, this again is a rat brain. And you take very thin sections, about a 0.1 millimeter slice, and then you evaluate on that section the presence of these proteins, whether it's a receptor or it's a transporter or it could be other things. But we'll stick with the receptors and transporters. So how do you do that? The steps are as follows. You take that slice and you put it in a solution that has radioactive ligand. And ligand is a fancy way of saying this is a molecule that has a specific affinity for that target. So it attaches to the target and it sticks to it. And it's got a radioactive tag on it. So when you incubate this brain slice in that solution and you wash it, the only thing that should stay attached are those things that are selectively interacting with that receptor or that transporter, and it's got this radioactivity piece to it, so it's sending out this radioactive signal. Then you take your brain slice after you've done this washing, and you put a radioactive sensitive film over the top of it, and you incubate it for a period of minutes, hours, days, depending on a variety of factors, so that the small molecules, the ligands with the radioactivity on it, will develop that piece of the film that's sitting right on the top. And you develop that film and you get something like this. Again, it's a rat brain. But you see dark areas, particularly like this dark area here, dark areas up here. This means whatever protein they were trying to measure, it shows up darker because that film has been developed in response to this radioactive ligand that's sitting right underneath it. So now you've got an anatomical view of where these receptors are, where these transporters or proteins are, and you've got a quantitative ability to determine whether there's a lot of them or there aren't very many of them. So you can quantitate whether there's a change in those proteins. And this density is quantifiable and allows you to do an assessment and a comparison between animals that have been treated or not treated, for example. So, for the most part, it's been used to assess these changes in receptors and transporters, and this is in response to drug use. Now let's take a look at one particular protein we're very interested in the drug abuse world. As I said, dopamine's a big deal for us in this biology. And the transporters that move dopamine are very critical in understanding the dynamics of that system. And that's what this thing is here. This is a transporter, a dopamine-specific transporter. So we're going to use autoradiography to assess this transporter. And that's what this is. Here are brain slices, and instead of using black and white, we colorized it to help identify areas that are hot. So these orange kind of areas, that means you've got a highest density, and then the colors go colder, that means lower density. So in the brain, and this is a dopamine transporter, a DAT ligand, a specific drug or molecule that's radio-labeled with an I-125. It attaches to the DAT protein, the dopamine transporter protein. And now you can see the distribution. Where do we have a lot of dopamine transporter protein? And you can see there are certain areas where that's true. We expose this animal to cocaine prior to exposing them to this ligand. And what happens? It blocks the DAT ligand. The DAT has no place, the ligand has no place to attach. So what does that tell you about where cocaine acts? It interacts with the dopamine transporter. That's what it does for a living. That's its function, is it changes the way the dopamine transporter works. That's how it increases dopamine release, is by fouling up the dopamine transporter. So now we understand this basic biology of the dopamine transporter and of cocaine in particular. And we've done this with every one of the drugs of abuse. So we really know at the molecular level how these things work and what they do. So it shows that dopamine or cocaine binds to the dopamine transporter, and it helps us to understand this mechanism. So the next piece was, if cocaine exerts its effect by sitting on dopamine transporters, what if we gave cocaine, or a person used cocaine for long periods of time? What would it do to the dopamine transporters? Would it change those proteins and alter the way they function? And the answer is yes. Here was an analysis. This was a brain analysis in a human. Cocaine users, open squares, and the dark squares are control subjects. And comparing in different brain regions, we find that the cocaine users have an increased expression of their dopamine transporters. It changes the basic way in which the dopamine homeostasis occurs. Consequently, it's going to alter the way all those dopamine systems are affected and their ability to function in the individual. When do we need to close?
About 10 to.
10 to? Oh, we can do this easy. Okay. The third one I want to mention is brain imaging. This is what's hot right now. Brain imaging in the last 10 years has just exploded. This technology has allowed us now to look into human brains. A lot of the work prior to this has been on animal models, although the humans showed that the animal models were very predictive. But now we can look right into humans without hurting them, while they're alive, while they're functioning, and we can ask some very critical questions about what's going on. And I want to focus specifically on this technology. This is functional MRI. How many of you have had MRIs before? So most of you know the technology. This is functional in that it's got a temporal resolution to it. It's not just one time point, but it's multiple time points that are separated by seconds. So you can literally see how the brain changes in response to prompting or an experience, or if you give them a drug. So you get this nice sequence that really gives you a dynamic understanding of what the brain's doing. So we stick somebody's head in a magnet. Usually the opening is smaller than this, so that you can just put the head in there. And functional MRI is based on a principle called BOLD, which is an acronym for blood oxygen level dependent imaging. So just quickly, without going into a lot of detail, we expose the brain or the tissue to a magnetic field, and this magnetic field is very sensitive, or is responded to by hemoglobin, and whether it's oxygenated or deoxygenated, the response is different. So we're able to distinguish between high levels of deoxygenation or high levels of oxygenation. Now, why do we care about that? Well, oxygenated hemoglobin generally is associated with new blood that's coming in. So if there's a part of the brain that's very active or it's been activated, we get a lot of blood flow into that brain area, which means there's a lot of oxygenated hemoglobin there, and our functional MRI system can pick it up. So we can actually watch where the blood goes in the brain and how it shifts between different regions. So it detects this blood flow, and the data say that neuronal firing correlates with blood flow. In other words, if I've got a lot of neurons firing in a certain area, the brain says, we got to get a lot of blood there to support this increased activity. With the functional MRI, I can see the blood go to this area that's firing or has been activated. This is just an illustration looking from the top down. Here's an individual where they've been exposed to language tests, and they colorize this so that you can see what areas of the brain have increased blood flow. And it turns out it correlates very nicely to those language areas of the brain. So there's some really clever investigation and research that's gone on, and that applies to the drug abuse world. So how's this science used? Well, let me give you an example. This was a paper that came out, it's been about 4 or 5 years ago now, using functional MRI. It was set up with two categories of methamphetamine abusers. In all cases, they were abstinent, so they hadn't used methamphetamine for several months. So they'd all been off the drug's cleared out. So this isn't a direct effect of the drug, but it's a persistent consequence of having used that drug. Then they gave these people a simple motor task, like the light turned on, they had to reach up and tap a lever. The light went off, comes on, tap a lever. So something that doesn't take a lot of brain processing, fairly simple, straightforward, and they did a functional MRI on that brain. Then they exposed them to a test that was more difficult. It required some decision making, where they actually had to be given information. The brain had to process that information, select a behavior that was appropriate. And they analyzed the brain, or the functional MRI of the brain, under those conditions. And then they compared the two in the same person. What brain areas were activated when they did the simple task? What brain areas were activated when they did the complex task? And here they have their two groups. In one group of these meth-abstinent individuals, they used exactly the same brain areas for both, for the simple tasks and for the complicated processing tasks. The other group, they used many more brain areas when they had to do the complicated cognitive behavioral tasks. And then they watched these people for the next 1 or 2 years to see who relapsed, because the relapse rate for methamphetamine addiction could be pretty high. They watched who relapsed. And which of these groups do you think was most likely to relapse? The one who used the same brain systems for simple tasks and complicated tasks versus the one that was able to recruit more brain systems for the complicated tasks? One? How many say one was more likely to relapse? How many say two was more likely to relapse? One was the more likely to relapse. It was almost 90% accurate in predicting who relapsed. And the way they interpret this, and obviously there's still a lot of work to be done, is that those individuals who damaged the ability of the brain to process complex information, they're still using very simple systems to make decisions, they don't do well when they're given the option to go back and use the drug, or do well in life in general, because they use the exact same strategies to decide or deal with all issues. Whereas those who still have all this reservoir capacity can now pull that into play. It's kind of like a computer. If a computer has very little capacity, it can't do complicated tasks. If it's got a lot of memory and a lot of capacity, it can do a lot more elegant things. And the same thing applies to the brain. So interesting use of the functional MRI. These areas are the ones that were the big difference in the ones who stayed off the drug. Decision making, was more activated in the more complicated process. There's a little area here called the insula that turns out to be important for tobacco use. That was different in the two groups. And the area of the brain that's important for compulsive behavior was different in the two groups. I mean, you'd almost predict this would be the case, but it underscores the neurobiology that seems to be important for addiction and dependence. And the last technology I want to mention is genetic technology and genetic analysis. How many know about microassays or microarrays? Ah, so this is a genetic institution. The microarray has provided a great tool for us to evaluate the underlying basis of addiction. So just quickly, for those of you that aren't familiar with this technology, it's a means of quantifying gene expression. And it goes something like this. This is Genetics 101. Your DNAs are made up of nucleotides and nucleosides. These things are transcribed into messenger RNAs. So these counterpart components are then used as a template to form proteins. So it's a way of assembling amino acids through the process of translation into proteins. And these proteins, as I've already mentioned, can be very, very important. Some of them can be receptors or transporters for the neurotransmitters. And of course, they cause or result in a biological function that's very, very important for us. Okay, that's the general process. You've all heard of polymorphisms, right? Polymorphisms are variant expressions of the DNA. All DNA doesn't look the same. Even though it's the same gene, it may have different forms because the pair, there's a base sequence that's altered, and it causes these variable expressions, which means you're going to get variable messenger RNA formed, which means you're going to have variable amino acid sequence, and your proteins are going to look a little different depending on whether you have a polymorphism or not, and that affects the ultimate functioning of that system. It would change the receptor, or it would change the receptor or the transporter, so they work less efficiently in this person if they've got a polymorphism that creates a problem for that protein. So what does that mean? Well, it means that we can use microarray technology to identify these polymorphisms. Now, we all have polymorphisms. Everybody has polymorphisms, but some of those get us into trouble. Especially certain clustering of polymorphisms can get us into trouble. And with a microarray strategy, we can identify very quickly where your polymorphisms are and whether this is going to be something that could make you vulnerable. In fact, an analysis, a retrospective analysis has been done, and they found that almost 100 polymorphisms, 89 to be exact, and these are all where the triangles are associated with different aspects of drug dependence and addiction. So that means those who have these polymorphisms are more vulnerable to various components of drug addiction. So there is a genetic piece to this, and we know now that almost 50% of vulnerability to addiction comes from genetics, or it's inherited. That's why we see clustering of addiction and dependence in families, good families. Families that are doing everything they're supposed to be doing. LDS families, parents that have temple recommends. It still shows up because there are these vulnerabilities that exist. So quickly, just to describe a really intriguing genetic discovery that was made, some of it at the University of Utah, another component was here in Iceland. They looked at this cohort of smokers. There was 10,000 in this cohort, and they were looking at nicotine, the subunit elements. Nicotine is a receptor, but it's got multiple proteins in it. It exists as an interaction between these protein subunits. So they looked at that, and they found that there was this polymorphism for one of these nicotine subunits that was associated with not only smoking, but lung cancer. And we know that smokers have a really high incidence of lung cancer. And we've always thought, well, it's because they smoke, and the smoking damages the lung, and then the lung becomes cancerous, and the disease develops. Well, from this study it said, well, that's true, but another piece of it is that this nicotine polymorphism makes the lung more vulnerable to cancer, whether you smoke or you don't smoke. It increases the likelihood of lung cancer regardless of what your smoking pattern is. Well, this group at the University of Utah looked at this same polymorphism, but looked at it in an adolescent context. And there's an interesting, some interesting data out there about smoking in adolescence. If you start smoking as before you turn 18 years of age, the likelihood that you'll go on to be a smoker as an adult is like 92%. I mean, it's much higher. 75% of these will try to smoke, but they won't be able to. They won't be able to give it up. And the earlier they start, the heavier the tendency or more likelihood they'll be to continue. If they start before they're 13, they only have like a 4% chance of not smoking when they're an adult. If they start when they're 14 to 16, it's a 10%. If they start as an adult, they have greater than a 90% chance to stop smoking. So that tells us something's going on during adolescence. So that's what this study looked at. It looked at this polymorphism in adolescence and smoking, and they found the following. If a person had this abnormal nicotine receptor and they started smoking as a young person, then they were much more likely to have problems. They were 5 times more likely to start smoking or to go on and smoke into an adult if they started smoking at the young age. However, if they had that polymorphism and they didn't start smoking until after 17, then there was no greater likelihood of smoking in this population. So what it says is that this is a polymorphism that has a window. An altered gene that increases susceptibility when you're young, but if you don't smoke when you're young, then its vulnerability goes away. It doesn't increase the likelihood that you will smoke. So these vulnerabilities are very dynamic and the biology is very complicated. Let's go through this quickly. My time's almost up. By knowing what these vulnerabilities are, we should be able to assess in people. We should be able to do a microarray, if you will, and say, oh, you've got vulnerabilities here, here, and here. You never get addicted or dependent if you don't use the drug. So if we can find those people who are most vulnerable and say, you really have to stay away from this stuff, because you're vulnerable to get into trouble if you get around it. Plus, we would hope that it would also give us better strategies for improving decision making. If we knew why the addiction occurred, now we can approach it and find better ways to treat these people. So let's just skip this, go right to the ending. That is, so what has technology taught us? Basic principles here. One is, we know now that drug dependence is definitely a biomedical problem, and it's got pathologies that look a lot like other diseases. We also know or have this knowledge, and we think that we can use it to prevent and diagnose and to do a better job in treating addiction disorders from the medicinal medical perspective. It's not just a morality issue of good versus evil, but it's an issue of health, it's an issue of sickness, and we need to take that approach with us as we approach and deal with these people. Thank you.